I'm very excited today to introduce the speaker, Jennifer Lippincott Schwartz. Jennifer is currently the Senior Group Lead and Head of 4D Cellular Physiology at the HHMI's Janelia Research Campus. And her group focuses on using live cell imaging to study spatiotemporal behavior of molecules and organelles across cells, across live cells. But before Janelia, she did her PhD at Johns Hopkins, where she looked at subcellular localization of lysosomal proteins. And then during her postdoc at the NIH, she looked at novel dynamic interactions between molecules and organelles and their structures, specifically the endoplasmic reticulum and the Golgi. And then she established her own lab at the NIH, which has pioneered the use of GFP, fluorescent proteins, for tracking molecules and structures in live cells. This is using FRAP techniques, photoactivatable GFP, super-resolution microscopy, to study a variety of phenomena, from protein dynamics to cell-cell fusion. Yes, so for all these contributions, she's been recognized by many institutions. So she is the president of the American Society for Cell Biology. She was an elected member of the National Academy of Sciences. She's an Academy of Arts and Sciences Fellow, and she was awarded the E.B. Wilson Medal of the American Society for Cell Biology, just to name a few of the accolades. So with that, I just want to say thank you to Jennifer for speaking today, and I'll turn it over to you. Thank you.
Thank you very much, Tara, for that really nice introduction. And thank all of you for joining us today. It's very exciting to be here. I really resonated with what Todd spoke about a couple minutes ago in terms of we're really being at a pivot point in biology, in science, with really I think what we can all describe as an AI revolution. Certainly, I've experienced it in my own world of imaging as a cell biologist. I will be telling you a little bit about the way we're trying to incorporate machine learning and AI into what we're doing in my presentation today. But I also want to just draw your attention to, at Janelia, we really recognize this need for computational power in analyzing data sets. So we're hoping, all of you who are postdocs or moving on to your next career after spending wonderful time here at Broad and the Schmidt Center, please consider Janelia, because we are basically launching a 10-year huge effort in AI, all aspects of AI in terms of experimental interrogation of various biological pathways to structural work. Okay, so today I'm going to be talking about exploring life structures across scales through imaging. And I want to get right to the thick of it, which is cells. Cells are the fundamental entities, structures that all life is comprised of. What's quite remarkable is the eukaryotic cell, which, as you can see, is this intricately organized system of many different organelles, can give rise to all of the life forms, complex life forms that we have on Earth. And what's incredible is that this system is conserved. The internal organization of the eukaryotic cell is conserved across all kingdoms of eukaryotic systems. A confederacy of subcellular structures that each have specific cell activities really define those membrane-bound structures that I just showed you in that EM image in the prior slide. This includes organelles like the ER and the Golgi that are involved in secretory activities of these cells. Organelles like the endosome and lysosomes that enable these cells to take up nutrients and other macromolecules from the outside to be degraded and recycled. Mitochondria, which are the energy powerhouse of the cell. The nucleus, where DNA replication and storage has occurred. Peroxisomes, where molecules are detoxified. And finally, this lipid droplet, where fats are stored. All of these organelles are interacting with each other, communicating with each other. And if we want to understand life's programs, we need to understand how these organelles are working in different contexts. So this is basically one attempt to really start digging into the dynamics of these organelles. Maybe if we had the lights down, it would be great for you to see some of these movies. But what we're doing here is simultaneously visualizing 6 of these organelles using a lattice light sheet multispectral imaging approach. One of the things that you can see from this movie is that these organelles are highly dynamic, and they are all interacting with each other in one way or another. Now, because this is a 3-dimensional movie where we have we have basically isotropic resolution across all of those different organelles. We can come in and map out how these organelles are interacting with each other at different times across any particular interval in the lifetime of that cell. And from that, we can create what we're described here as a matrix representation of the organelle interactome. Basically, the extent to which these organelles are interacting with each other over time. And what's interesting is that even though these organelles are highly dynamic, where you can see these contact sites forming and dissolving dynamically, at steady state over long periods of time, you have a conserved type of interactome among these organelles. And those interactions are playing critical roles for these organelles to communicate not only lipids, but also small metabolites, as well as ions like calcium. Now, we can go from the micron level, which that lattice light sheet movie was showing you, to the nanoscale using electron microscope approaches. What you're looking at here is a whole cell EM volume across a HeLa cell, where we are acquiring, we're basically milling across this cell at 4 nanometer slices, if you will. So we can, and then we're imaging each one of the regions that we've milled off with structure illumination, with scanning electron microscopy. So when you do that, you now have the ability to basically 3-dimensionally interrogate the entire cell at the nanoscopic level. So this is a 1 cubic micron region of that HeLa cell that you just saw. So it's the center of the cell. You can see there's the centriole. What we're looking at is we're just scanning through this 1 cubic micron region. You can see all of the individual slices, basically, that we imaged. And if we then reconstruct all of the objects that we see in that cubic volume, you now have the ability to define many cellular structures, including microtubules, the Golgi apparatus, the endoplasmic reticulum. This is the nuclear envelope. This is the nucleus. All of these structures become visible at this nanoscopic level. Now, this was not an easy task. Each one of the 4 nanometer slices is only a tiny part of any particular structure. You have to stitch them all together, and that requires hand annotation. So basically, it took a single person 7 to 10 days to reconstruct this 1 cubic micron of all of these membranes and the way they lay out over this volume. So that led to an immediate problem. You know, the cell is actually 7,000 times this size. If you want to see all of these organelles at this scale of imaging, we estimated a single person would take you 50 years to do this type of manual segmentation. So instead, at Janelia, we set up a team to do this using machine learning. Basically, individual annotators would go in and annotate small subparts of the cytoplasm, and then feed that information to a a larger machine learning algorithmic program, which then would basically automatically search for the objects that it had been trained on. So basically, as a consequence of that, we could really reconstruct very quickly, in this case, up to 32 different structures within this HeLa cell. And we could compare, as a consequence, we could do it with different cell types. You could compare volumes, organization, the way these organelles are contacting each other across many different cell types. And currently at Janelia, we're doing this in much larger volumes, whole tissues from various organisms to look at, interrogate cellular organization at this nanoscopic scale across cells within particular tissues. Now, this all sounds really cool, but it turns out that when you look at these images, there are many structures that are unrecognizable. Like, what the heck is this? And there's also hard to find structures. You know there should be a structure, but you don't know what it looks like. And so how are you going to find it in these data sets? And so that's where basically we're sort of trying to push new approaches. So here's an example where just of a weird looking structure that if you were going through these data sets, you could see. Okay, I don't know what that is. That doesn't look like a Golgi, it doesn't look like a lysosome, doesn't look like an ER. The only way that we have been able to annotate all of the structures that I showed you is because we already know what those structures look like. We have a hypothesis for what they look like. This is something we don't know what it looks like. And the problem with our traditional supervised learning strategy is that the way we go about doing our analysis is we manually annotate things that we're interested in, and these things have to be fairly abundant in order for the machine learning program to learn it. So basically, you annotate something you're interested in, ER, mitochondria, lysosomes, and then you use a machine learning to basically interrogate the entire sample. And it will very nicely pull out all of the objects that look like a mitochondria or look like an ER. But you cannot train it when you see a weird object that's infrequently, it's not abundant. So it doesn't, you can manually annotate it, but you don't have enough examples of it for the machine learning to be able to see it within the system. So the approach that we're currently trying to do at Janelia is a self-supervised learning strategy where instead of manually annotating and creating training data for machine learning algorithm, we're basically having the computer train on the entire data set. Basically put the entire data set into its head. That takes a lot of machine time intensity, but that's better than having a human trying to do many different objects simultaneously. But once you have the program where it has the entire data set sort of at its fingertips, you can then query it for different objects. And when you do that, it can pull out weird-looking structures that are at very low abundance. Now, once you've been able to identify where those structures are across a cell or across cells under different perturbation conditions, in this case, this structure is being generated when we stress cells, you can then come in and apply other approaches. In this case, we're using correlative electron microscopy to show that this structure is actually an autophagosome that has engulfed an ER exit site. Now, this cryocorrelative electron microscopy is really, really important, we think, for really pushing our understanding of the organization of these cells, because it's also valuable for trying to look at the structure of hard-to-find structures. One of those hard-to-find structures are sites where all secretory and membrane proteins exit the ER. Secretory and membrane proteins are synthesized in the ER, and they exit the ER at very specialized sites that are created through this machinery, COP2 coat machinery. And the COP2 coat in vitro can create vesicles. So everybody thought, oh, well, let's look for vesicles. Maybe we can see COP2 vesicles in these big EM data sets. Not so easy to find. But if you do cryocorrelative, basically you introduce a fluorescent probe for a component of this COP2 coat, which you know is defining the ER exit site. You can then, at the light level, image where all those exit sites are localized in this cell. And then what you can do, and this is, we're imaging at cryo temperatures. So we can now take, and this has been high pressure frozen, this cell. So we can now take this cell and do our FibSim milling through the entire cell and interrogate individual areas where we have the COP2 signal. And when we do that, what you can see, this is a little cubic volume here. We're going to go up and down through the different 4 nanometer sections that we've acquired through scanning EM. And here's the spot where we're getting this Sec31 signal, or I'm sorry, Sec23 signal. And if we then interrogate that spot and basically stitch together all of the individual images, we can reconstruct what these ER exit sites actually look like. And surprise, surprise, they are not vesicles that are sort of randomly budding off the surface of the ER. They are very discrete structures. This is the endoplasmic reticulum. There's a tight neck where this Sec23 coat complex is operating, and then splaying out from that neck is a membrane labyrinth, sort of like a grape-like cluster of membranes that have a very characteristic diameter of these sort of grape-like tubule extensions, as well as the entire structure. So once we have knowledge of what this looks like, we can then go back into our data sets to be able to find ER exit sites in any cell, now that we have the signature of what this structure looks like. Okay, in the remaining few minutes, I want to move to the atomic scale. So we've looked at the micron scale, we just looked with EM at the nanoscale. Now we're going to go to the atomic scale and how imaging is dramatically improving in this area, but also this is a total area where we definitely need help through computational biologists and machine learning. Okay, so this is an archaea cell, but like every other cell on Earth, it is loaded with these small little structures that we call ribosomes. So this is just highlighting ribosomes in a archaea cell. But they are found in every eukaryotic cell on the planet because they are what drive protein synthesis. So the ribosome is the core machine of translation. This is how proteins are being made. So it's a macromolecular complex that is comprised of a large subunit, a small subunit, and it's bringing together mRNA that will be decoded and converted into a polypeptide chain as a result of recruitment of these tRNAs that are charged with various amino acids. Now, this structure has been studied for decades in terms of what it looks like at the atomic scale. What we're going to try to do is to ask whether we can actually identify these structures in an intact cell that's been cryofrozen and basically get enough snapshots of this structure to actually make a movie of translation at the atomic scale. Okay, now this has at some level been done from in vitro, from prior work where various structures of the ribosome have been identified through single particle averaging. What that has shown us is that the ribosome indeed undergoes conformational changes during this elongation cycle. The cycle for moving the mRNA through that ribosome to allow a polypeptide to form. What we want to do is to get a lot more of these intermediates and stitch them together to see if we can see a movie of this process. So the approach that we're going to be taking is called high-resolution template matching. And I want to shout out Peter Rickauer, who was one of the pioneers of this approach, together with Winfried Denk and Nico Grugier at Janelia. Basically, what this approach does is it uses a high-resolution, noise-free template of a macromolecule, and you search for that macromolecule exhaustively in an image that's been acquired with minimal electron beam exposure. Now, key things here. One is we have lots of high-resolution templates. I mean, the field is exploding, AlphaFold, millions of templates. Let's find them in a cell. Can we see these things in a cell? So this is what this approach is going to be doing. Now, the approach is not dependent on the abundance of a target or averaging, which is what traditional single particle cryotomography approaches are using. Those approaches basically look at abundant structures, pull them out, and then do single particle averaging to look at various states of that structure. We're not going to be doing that. We're going to just look at all of the structures we can see using the template. And then basically, we're going to use the high-resolution signal in the noisy images to gain insight into the structural states of the molecules. So here's the approach that we're taking. Cells are grown on gold grids. They are then high-pressure frozen. And so you can see here individual cells that are on that grid. These are the cells that are going to be examined. And we're going to examine them with a single image on our Krios electron cryo-EM system. Now, this is an image from the periphery of the cell where the cell's about 200 nanometers thick. So we are viewing everything. Our electron beam is moving through that entire 200 nanometer section. This is the image that we get. Okay, so you're going to say, wow, what kind of image is this? I don't really see much in this image, which is exactly what I told Peter when I first saw this. But he said, look, the key thing here is that this image has information. We have not destroyed information with the electron beam. One of the problems with cryotomography is you're hitting your sample many, many times with your electron beam. Every time that electron beam moves through this tissue, it destroys information. So we have one image we've acquired right here, and we're going to search it for a target. And the target we're going to be looking for is a ribosome subunit. This is the large subunit of the ribosome. We're going to search for it in every possible orientation that we can find. This is what this ribosome template would look like in our EM image. If electron beam is moving through this, this is what this thing's going to look like. So we're going to simulate this structure. We're going to rotate it in every orientation and see if we see any cross-correlation with anything in our real image. And sure enough, we get hits. So that spot right there is precise in here is where this structure is. We are very confident of that. This is the signal to noise showing that in this image right here is this structure. Now, we can do this across our entire data set. And so what we're doing here, and we can do it not only for the large subunit of the ribosome, we can also do it for the small subunit, in particular, the body and the head of that small subunit. So each one of these are different molecular weights, as you can see. With that large subunit, we have much better signal to noise because it's a bigger target. We go down to the body, small subunit body. We still see very significant hits, signal to noise. The head is a little bit harder. It's only 350 kilodaltons, but we can still get signal. And we're doing this independently. These are separate parts of the ribosome that we are trying to look for. And this is just a decoy, where we just put in a 2,000 kD structure that we know is not in this cell, and we don't see anything. So this is where all of these subunits are localized in that image. Here are all our peaks of correlation, cross-correlation, and if we fit in the position of the large subunit that we're detecting independently of the head and the body, you can see where all of these guys come together in complete ribosomes. Some of these ribosomes are not fully assembled, and we can actually see that. Others are fully assembled. But we're seeing all of the ribosomes in this data set. Okay, well, what can we do with this? Well, and this is just showing how these things are arranged. I'm just going to move very quickly to, you can just correlate it onto the image. So what can we do to this? Well, during translation, the key, first off, the ribosome is a machine. And the way it works is by rotation of the large subunit relative to these small subunits. The small subunits are swiveling, the large subunit is rotating. The swivel motion is moving the transcript forward by one codon during polypeptide synthesis. So we want to look at all of the ribosomes that we've seen. We want to know what rotation swivel angle of these subunits each of those ribosomes are in. And that's what this is showing right here. So this is a plot where basically we're looking at the rotation. We're positioning all of the ribosomes in a swivel to rotation plot. Each one of these dots to the far left is an individual ribosome in that data set where we can map out what its swivel angle is relative to its rotation angle. And we can then fit all of those ribosomes in a path. And we're just arbitrarily dividing all of these guys up into 10 different states of that path, which reflects the elongation cycle. So basically, we can, from the data, basically look at the populations of the ribosomes in each one of these different states. We can also look at their relative abundance, and we can interrogate what these states are during this elongation cycle. Now, some of the interesting things that have come out of this is oftentimes individual transcripts shown here, mRNA transcripts, have many ribosomes associated with them. These are called polysomes. That enables efficient translation of this mRNA. What we found was that the ribosomes that are attached to this mRNA, which we call polysomes, many of them are actually in the same state of this elongation cycle path. That was very, very interesting to see this. You can see that there are many of these adjoining ribosomes are all in the same elongation stage of the elongation cycle. This is not colliding of these ribosomes, because we can actually go in and interrogate the structure of these ribosomes that are neighboring each other, that are in the same cycle state. They do not fit with what has been described from in vitro studies of ribosome colliding. We're seeing a very different type of interface of contact between these ribosomes. So what that's led us to speculate is that these ribosomes aligned along the mRNA are actually synchronized in terms of their movement. Much like if you were on a rowing team, if you're going to row effectively, you've got to be synchronized with these motions, and that's what we think these ribosomes might be doing. Okay, I only have a couple more minutes, so I'm going to go really fast to get to the movie. But basically, one of the things you can do is, and this is really important, is you can reconstruct molecules that are around these ribosomes at different states of the elongation cycle. And this is, I'll just show you this right here. This is a ribosome that we're looking, whoops. So each one, all of these structures that you see in gray here and that are color-coded represent molecules that we have been able to see associated with these ribosomes that we can see at different states of this cycle. So these were not we were not interrogating, we're not doing template matching on these structures. They came out because they were electron densities that were associated with the ribosome that we had identified. And this is just showing we can actually see mRNA, we can see the mRNA tunnel, we can actually see P-site bases in this system in terms of the resolution capacity. Okay, now I want to just quickly show you a movie where we're trying to stitch all of these guys together. We have 41 different ribosome states that we can fit in this elongation cycle pathway. And so what we're going to do is just, we can align them based on what we know in terms of the changes in the conformation of the ribosome, and we can then play it as a movie. And that's what we're doing right here. So we have, this is the large subunit. This is the small subunit, the head and the body. These are associated stuff. Right here, tRNAs, synthases that are carrying amino acids into this pocket where a polypeptide is going to be pulling off from it. These are other molecules that are associated with the ribosome during this elongation state. So this clock right here is just running to show the whole cycle. So this is just different types of motions that we're seeing. This is 20S RNA that's basically showing rigid body motion. It's tightly associated with that large subunit. We can also see a interbridge protein that's undergoing a type of bending motion that's never been seen before. But we can pull it out from these data sets because we're basically aligning all of these ribosomal states in a movie. And then finally, we can look at these tRNAs, synthases, as they come in and move across these the EPA sites for stitching together the polypeptide. So this is just summarizing the outlook for this approach, as well as other cryotomography approaches. This can be very much integrated together with the system. I want to point out that I think it's super exciting in terms of what we can do with structures that are coming out, AlphaFold, other types of structures that we know. It has the potential to be used in this type of system to be able to more fine-tune the way these molecules are actually behaving within a cell. So with that, I'm going to end and thank all the people at Janelia who've participated in this. Peter Rickauer, I want to call out again, is the guy who was critical for this high-resolution template matching. But other people in my lab have been heavily involved in all of this imaging at these different scales. None of this would happen without incredible support teams from cryo-EM to light microscopy to computer support, as well as other group leader interactions at Janelia. Thank you very much.
All right, I think, oh yes, okay, we have time for one question, maybe.
I'm going to really use it as a long question. First of all, this was a really fabulous talk and a great way to open this meeting. Very inspiring what can be done now. You know, in an attempt to show how cool everything is, I'm being the forever skeptic. Are always like, how accurate is this? Does it know when it fails? Where does it fail? And again, to this wonderful audience of brilliant trainees behind me, where do you need them?
Okay, so I'll tell you exactly where we need you. So, you would think that we have a lot of X-ray crystallography structures. They've been purified, crystallized. If you take those structures and you try to find them in our data set, most of them we cannot find. What does that tell us? It tells us that structure is not the structure that is the abundant form in this intact cell. So the issue is, how do we go about trying to use the information from these cryo images. And AlphaFold is the same. AlphaFold has lots of parts of it that you're not going to see here. The issue is, can we find parts of these structures that we are pretty confident are going to be stable, are going to be static? And then what we can do is we can actually just use that as our template. Forget the rest of the protein. Let's find that as a template. And as I showed, I'll just quickly go back here, sorry, because this is pretty significant. We've done this here. This is the ribosome, okay, the large subunit of the ribosome. If we scale it down to only 700 kilodaltons, we basically shaved off a huge part of this ribosome, and we're going to search for it within our sample. We can find it we find it, and then we look at the reconstructed density, we get the ribosome. And we can do this down to, this right here is where we've got a 700 KD target, where we're successfully doing that. In comparison, cryotomography and SPA approaches, you don't get anywhere near that in terms of identifying the structure within an intact cell. So we really need help from people who can sort of help us. Well, first, the compute time is huge. We need to figure out ways to really make this refined. To do the searching took us 2 months to search, pulling out those ribosomes in just one simple case. So having better, computational sort of pairing, et cetera, could speed this up. But also, being able to understand what these structures, how to deal with these structures, I think would be really important. Great question.
Okay, great. Thank you. So unfortunately, we will just do the one question, and I'll pass it over to Sebastiano to introduce our next speaker.